Hey everyone, here is another series of the test and discussion. So, as I said before in the earlier episode, the test link is given below in the description box. So, just whoever is viewing this, just go to the description box, check out the link, and solve the 5 MCQs via the test link. After which you just listen to the discussion part. Okay. So, this is how the T&Ds will go on, and it's mainly aiming or targeting the entrance examinations like NEETPG, FMGE, and INICT, right? So let's move on. So the first question is hepcidin inhibits what? Hepcidin inhibits absorption of cobalamin or transfer of iron into enterocytes or folic acid synthesis or respiratory oxidase. So what does hepcidin inhibit? So in first place, what you should know is hepcidin. From where does it come? Hepcidin is involved in iron metabolism. Okay. So if you know that, you will be able to rule out the options. So it inhibits the transfer of iron into enterocytes. So that's going to be the answer, right? So this is the option B is the answer here. So what you should know about hepcidin is that hepcidin is involved in iron metabolism. Iron regulation is mainly involved in the iron regulation. So iron being a one-way element can be regulated at the level of absorption from the intestine. Okay. So here, whenever iron stores are adequate, then hepcidin will play a role, will be a negative regulator by inhibiting the iron absorption from the intestine. So that's the point, that is the learning point here. So as you can see in this picture, you'll be able to appreciate that the food iron has heme iron and non-heme iron. Heme iron is directly taken inside the intestinal cell via the heme transporter, whereas non-heme iron, the ferric part is getting converted to ferrous part, and the ferrous iron is what will be taken inside the intestinal cell via the divalent metal transporter 1, which is present along the apical membrane of your intestinal cell. So, which is converting the ferric to ferrous, that is by the duodenal cytochrome B. Yes. So, in most of the individuals, if the duodenal cytochrome B is absent, then for them, the conversion of the ferric to ferrous becomes deficient. So, in that can be a reason of iron deficiency anemia. So that is why we always suggest to give, a lemon juice or an orange juice along with the iron supplement. That is to enhance the conversion of this ferric to ferrous. Okay, so that it can be easily absorbed via the ferrous form. So after it gets inside, it either gets stored in the form of ferritin, yes, ferritin, or it is getting transported to the bloodstream. So along the basolateral membrane, you have something called as the ferroportin, through which your iron gets out. And again, the hephaestin converts the ferrous to ferric form, and the ferric form is what is the transport form, right? So ferric gets combined with the transferrin in the circulation and gets transported. All right, so to the liver or to the erythroid marrow. So ferric gets transported in circulation. Ferrous form is what will enter into the intestinal cell. Okay, so this is what you have to understand here. Now, where does this hepcidin come? Hepcidin is here, which is synthesized by the liver, and it inhibits the ferroportin. Okay, so whenever you have adequate iron stores, then this ferroportin is inhibited so that iron doesn't come out of the intestine only. Yes, so this is how the hepcidin acts as a negative regulator of iron. All right, let's move on to the next question. Ochronosis is due to accumulation of what? Yes, the options being homogentasic acid, xanthurenic acid, fumaryl acetoacetate hydrolase, glyoxylate. What do you think is the answer? So just take 2 seconds. What is the answer here? So it's a very, very, very important question. Ochronosis is due to accumulation of homogentasic acid. Yes. So what's the condition here? It's the inborn error of amino acid metabolism, which is the alkaptonuria, right? Alkaptonuria is due to the deficiency of homogentasic acid oxidase enzyme. So if that gets deficient, then the precursor or the substrate of the enzyme, which is the homogentasic acid, gets accumulated. If homogentasic acid gets accumulated, it gets auto-oxidized to benzoquinone. That benzoquinone is what, this what is responsible for this black thing. Okay, so the black discoloration. So black discoloration of your pinna or IV discs. Okay, so these wherever the homogenetic acid gets accumulated, so that is what is called as the ochronosis. All right. So let's see. This is the answer. So homogenetic acid is the answer here. So as you can see, this is a enzyme that is involved in the tyrosine metabolism pathway. Okay, so here the homogenetic acid gets converted to mallyl acetoacetic acid with the help of homogenetic acid oxidase or dioxygenase. If this enzyme is deficient, that leads to the accumulation of homogenetic acid, and this condition is what is called as the alkaptonuria. Homogenetic acid gets oxidized to benzoquinone acetate. And that is what is the ochronotic pigment here that is responsible for the urine turning black on standing. Okay, so urine, whenever it's collected, it's straw colored only. But when you leave it for some time, when it's exposed to air, it gets auto-oxidized and becomes this ochronotic pigment and it becomes black in color. Similarly, as you can see in the conjunctiva, in the pinna of the ear, but also in the bone. So this is what is called as the ochronosis. Is it fine? So this is alkaptonuria is one of the important topics for the entrance examinations. Moving on to the next question. Hormone that's predominantly secreted after the 14th day of by the endometrium is what? That's going to be, yes, it's very, very obvious. It's going to be progesterone, is it? So let's see the cycle. So what the menstrual cycle, the cycle is of follicular phase and luteal phase, right? So along the 14th day is the period of ovulation, is it? So 14th day is the period of ovulation. So where you get the LH surge, which is enhancing this LH surge, which is causing the LH surge is what is the estrogen. So estrogen getting peaked up, that is stimulating the LH surge. So this is what is called as a positive feedback mechanism here. Okay, so estrogen getting higher, that is leading on to LH surge. So this is another important point which you need to remember in this topic. So LH surge along the 14th day. Then after this, you have what is called as the secretory phase. You have the formation of the corpus luteum. And which hormone is more here? It is the progesterone that is getting secreted more. Yes. And the first is the follicular phase, and you have the FSH, the follicular stimulating hormone, that stimulates the formation of follicles. Then 14th day, you have the LH surge, that's the ovulation. After which you have the secretory phase, synthesis of, the formation of corpus luteum, and then the synthesis of progesterone. Yes, so these are the points that you need to remember in this. Yes. Next is mode of inheritance of Leber hereditary optic neuropathy. So what disease is it? Yes. So whenever you find, difficult names, what is it? It's going to be Leber hereditary optic neuropathy. You would have seen this abbreviation, is it not? So that's going to be mitochondrial DNA damage disorder. It's not a DNA repair mechanism. So you need to know what are all the diseases that come under DNA repair mechanism. For example, nucleotide excision, base excision. Yes, so you have xeroderma pigmentosa, M-tooth syndrome, yes, ataxia, telangiectasia, Bloom syndrome. So all these things you have to know. That belongs to DNA repair mechanism defects, right? And then what are the lipid storage or the lysosomal storage disorders? What are the sphingolipidosis? What are the glycogen storage disorders? So you have to understand the names. So as we discuss different questions on these, we will discuss those diseases also. So as pertaining to this question, it's the mitochondrial DNA damage disorder is what is the Leber hereditary optic neuropathy. So what you should know about is what are the different diseases, mitochondrial diseases. One can be this Leber hereditary optic neuropathy. You have Leigh syndrome. You have MELAS. MELAS, the abbreviation itself says ME is for the mitochondrial encephalomyopathy, LA for lactic acidosis, S for stroke-like episodes. And then you have myoclonic epilepsy associated with red ragged fibers, then Kearns-Sayre syndrome. Okay, so as you can see, melas, the features are the name only. Features are those which are present in the name of the disease, melas, right? So you have the seizures, you have headache, you have vomiting, hearing loss, and all, limb weakness, stroke-like episodes. Yes, so you have lactic acidosis. So this is about melas. And Leber hereditary optic neuropathy is. Yes. So Leber hereditary optic neuropathy, sorry, that's the most common, and you have, the maternal inheritance. So all the mitochondrial diseases are predominantly maternally inherited. Okay, it's maternally inherited. So that's one important point that you need to understand. And you have Leigh syndrome, you have subacute necrotizing encephalopathy. Kearns-Sayre syndrome, it's again, you have, ophthalmoplegia here, retinopathy, all eye manifestations, and you have cardiac conduction block. And myoclonic epilepsy associated with red rag fibers. Red rag fibers is what you see in the muscle biopsy along with epilepsy, myoclonic epilepsy, ataxia associated with it, right? So these are the different mitochondrial diseases, right? Let's move on to the next question. Bilirubin in serum can be measured by. So what are these tests and what is Vandenberg reaction? So that's what is important here. So whenever you collect the blood, you separate the serum, and in the serum is what you measure the bilirubin. So be it direct bilirubin, indirect bilirubin, or total bilirubin, the reaction that is used to estimate the level of the bilirubin is going to be Vandenberg's test. Yes, so higher end you will use it to quantitate the bilirubin levels, but if you want to know qualitatively also, the Vandenberg reaction is what is the basis, right? So let's see, what is this Vandenberg reaction? So that's a diazo reaction. Here, the reagent is going to be diazosulfonelic acid. So diazosulfonelic acid is a reagent. If you are going to add it to the serum, yes. So if the serum, then it gives you a color. Yes, if it gives you a color, then directly it is giving you a color, is it not? So that will tell you that it's a direct bilirubin or the conjugated bilirubin. Okay. So diazosulfonelic acid, which is a bilirubin, if it gives you a azobilirubin complex, that's a purple color, then directly if you find this, then it's going to be the direct bilirubin. Okay. But if you're going to add methanol to it, after which you're going to get the color, then that's going to be indirect bilirubin or the presence of unconjugated bilirubin, right? Then there is something called as a biphasic reaction. So what is this biphasic reaction in the sense? First, with the reagent, you're adding the serum, it is giving you color, but upon adding, methanol, it is giving you the color intensifies. So in that case is that's the biphasic reaction. So which means both the direct and the indirect bilirubin is present in the sample. So hope this is understood. Yes. So diazole reaction, which is the reagent is going to be diazotized sulfonelic acid with direct bilirubin. It directly gives us a purple color. With indirect bilirubin, if it is present in the sample, then only upon adding a methanol, we will get a color. So that indicates the presence of unconjugated or the indirect bilirubin. Biphasic reaction is where the serum has both the direct and the indirect bilirubin. In that case, first I will get a color directly, but when I add methanol, the color intensifies, right? So that indicates the presence of both the conjugated and the unconjugated bilirubin. All right. So these are the 5 questions. So hope this was useful. So we'll continue it. So before watching the session, you should have gone to the test link. You should have given the test. Okay. Yes. Thank you all. See you with another episode.